Greetings everyone. Today we embark on a journey extracting DNA from strawberries. But why strawberries? Well, it turns out that strawberries not only delicious, but they're also DNA powerhouses. They happen to be octoploid, meaning that they have 8 copies of each set of chromosomes, with a total of 56 chromosomes. Now enough talking, and let's prepare our ingredients. We need 2 fresh or frozen strawberries, 2 tablespoons of dish detergent, 1 plastic bag, half cup of isopropyl alcohol, 1 tablespoon of salt, half cup of water, 2 plastic cups, 1 coffee filter, and 1 wood stick. The dish soap is playing a crucial role in breaking down the cell membrane surrounding the strawberry cells. This process is known as lysis, releasing cellular components including DNA into the solution. Next, we need a plastic bag. It provides a contained environment preventing contamination during the mashing process. And now let's talk about isopropyl alcohol or isopropanol, which is C3H8O. This alcohol plays a crucial role in precipitating the DNA. Since DNA is not soluble in alcohol, it precipitates out and DNA becomes visible. Moving on, we need 1 tablespoon of salt. This salt helps maintain the structure of the DNA by neutralizing the negative charges on the phosphate groups of the structure of DNA. But how does this happen? To understand the process, let's dive into the structure of DNA. DNA is a double-stranded molecule composed of nucleotides. These nucleotides consist of a deoxyribose sugar, phosphate group, and a nitrogenous base. The phosphate group carries a negative charge. Due to these negative charges, DNA strands naturally repel each other. So when we add salt in NaCl to the solution, it dissociates into sodium ions and chloride ions in water. The positively charged ions plays a crucial role in neutralizing the negative charges on the DNA strands by surrounding the negatively charged phosphate groups of DNA, making neutralization. Neutralization is crucial for successful precipitation, a process where cold alcohol is added during DNA extraction, making DNA visible for observation. Salt helps to remove proteins that bound to DNA. It also helps to keep the proteins dissolved in the aqueous layer, so they don't precipitate in alcohol along with the DNA. We also need a half cup of water, as it helps as a solvent for the DNA extraction solution, and it helps in dissolving the salt and the detergent. In addition, 2 plastic cups to hold the filtered strawberry mixture during the separation process. In addition, 1 coffee filter to extract the solution from the strawberry particles. And finally, 1 stirring rod to help moving the DNA from the solution. Firstly, we need to put our alcohol in the fridge because lowering the temperature enhancing the DNA precipitation. Firstly, we pull off any green leaves on the strawberries. And then we put the strawberries into plastic bag. Here our plastic bag. We put the strawberries inside. 1 and 2. We can use fresh or frozen, same as we said. Then we seal it. And gently we smash the strawberries for about 2 minutes. We smash the strawberries to open the cells and release the DNA. Then we have to mix together 2 teaspoons of detergent, 1 teaspoon of salt, and half cup of water into the plastic bag. We mix 2 teaspoons of dish detergent. First one. Here. and then second one. After the detergent, we have to put 1 teaspoon of salt. We would use another spoon because this spoon have this detergent. One teaspoon of salt, then half cup of water for our mixture. And this will be our DNA extraction solution. Now we will pour the DNA extraction liquid into the bag with the strawberries. And our bag. We close the bag. Seal it. And then for extra 1 minute, we smash our strawberries. Now let's open the bag and try to pour the strawberry liquid inside our filter. Then have to take care. Otherwise, we'll make a disaster. Yeah. Now we gently try to squeeze our liquid into the cup. And now let's try to pour our alcohol. We have just to wait for some seconds until our DNA is developed. And then with our wood stick, we extract our DNA. And here we have it, our DNA. I can save it here in this test tube, just as memory. And here we have it, our DNA. Dear viewer, last but not the least, don't forget to subscribe to our channel for more exciting videos. And if you got some strawberries, it's probably best to enjoy them the delicious way while we handle the experiments for you. Thanks for watching. Now, my poor friend, I understand why you are miserable. Even strawberries have more chromosomes than us.